Yeah, next we have Matthew Hartley from EMBL-EBI. He's going to be talking to us about scale, essentially. That's what they do there. Towards 1 billion images is a great title. I like it.
And can you hear me still? No response. Hello? Good, right, you can hear me. Super. Okay, hi everybody. Really sorry I can't be there in person, but it would be lovely to see you all, and I can both see familiar backs of heads and hear familiar voices. But greetings from sunny Hinxton, where EBI is situated. And for those of you that aren't aware, as Eric said, we kind of do scale, but we do scale in biological data resources. So among other things, we run databases for lots and lots and lots of different types of biological data, everything from kind of sequences to protein structure to chemical data. But the part that is dear to my heart, and I guess also professional life, is imaging data. And my teams run our two big imaging databases, the Bioimage Archive and Empire, the electron microscopy public image archive. And I will tell you a bit about those. Imaging, as everybody's aware, is really lots and lots and lots of different techniques, all of which are used to look at living systems, different length scales, different tissue sizes, different depth, fixed or live tissue, and they all produce really, really, really different types of imaging. And this is one of the big problems we have in data management. This is some subset of the thumbnails from different images that we have in our data resources, and you can see lots of different modalities, lots of different length scales. We absolutely have histology imaging here, and therefore if everybody wants to work on better compression mechanisms in OME-EBI, that would be fantastic, because this is definitely a problem for us as well. But we have a lot of very, very, very different things. We are specifically running deposition databases. And the point of deposition databases is, in a sense, we're brokers. We're not really publishing our own data. We are taking data from the outside world, where people come and they give it to us, and they typically do that when they're publishing a scientific manuscript. Not always. We do take data at other times. We take it, we get some of the minimal metadata that Kaya introduced, and we hold it partly to make the data more reproducible, to make scientific results more reproducible by giving access to the data on which they're built. And that's the big motivation for journals. That's why we get the emails at 5 p.m. on Friday from people saying, hey, my journal wants me to submit data, help me do it now. But we also take the data so that we can give it back to do interesting new things like image analysis methods development, like training humans as well as computers, but also finding new and interesting science out of the data. In terms of kind of current scale, the Bioimage Archive has little over 900 individual accessions, entries, roughly corresponds to the number of manuscripts for which we're holding data, not quite. And we get about 1 new entry a day. We crossed the 1 new entry per day threshold late last year. This graph is not cumulative, so this is showing the yearly number of depositions we get, and it's rising quite rapidly. So last year we got 300 new depositions. The year before it was 190-ish. That's going up. What's also very, very clearly going up is the amount of data incoming. So this is rising very much exponentially. But it's still actually not huge in terms of the magnitude of the resources we run generally. So if I put it into the context of EBI's data resources, the Bioimage Archive is this maroon dot here, clocking in at about 800 terabytes when we measured this last year. It's still a couple of orders of magnitude under the sequencing databases. So shotgun nucleotide sequencing is still at the kind of bit under 100 petabyte mark. There's quite a lot of headroom for us to grow, which is good, but we are growing, and the imaging is actually the fastest growing data type at EBI. So of the top 3 fastest growing resources, all 3 are imaging, if you count EMDB as an imaging resource. Data comes from all around the world. It's an international database. The most represented countries, if you look at the organization of the submitters, are the UK, USA, and Germany, who I think very well represented in the room where you are. But there's a long kind of tail of the distribution, and we have now, I think, I haven't updated this slide in a little while, somewhere between 45 and 50 different countries of origin of people submitting data to us. On the presentation, on the data outside, we have hitherto been kind of, Kaya mentioned, people don't really want to see just flat lists of files and metadata. And so far that has mostly been what we have presented, but actually I am now going to tab from the presentation into a web browser. And this is the alpha version of a new website, which I'll say more about a bit later in the talk, where instead of presenting the data in a much more sort of dry listing format, we actually show images, we show representative images for individual data sets. When we pick one and look at it, we can see metadata for that data set, both descriptive generally and somewhat more in depth following the EMBL guidelines, and more on that in a moment about both sample and specimen and acquisition metadata. And then when we drop down to the level of individual images, and we do have now, at least within the alpha site, we have landing pages for images, not just for whole studies. We're at this point into an embedded viewer. This is Bizarre from HMS, and you can do the nice things that were mentioned earlier in terms of being able to kind of zoom in, which you really want to do from histology down to the individual cellular level up to tissue level. We also present, and I didn't click on it there because it's nicer to pick an example where you'll see the 3D. One of the beautiful things about doing this using OMEs behind the scenes is that as well as use one viewer, so in this case Bizarre, this is 3D data, and you can kind of see individual plane slices, but it would be really nice to be able to see a little bit more about what the tissue shape looks like there. And we can, because we can use the kitware viewer, which does a really fantastic job of rendering this data with no pre-meshing or anything like that in the browser. And that's really nice for us because it means we don't have to do extra work beyond the initial conversion, staging the data to S3. Final thing I'll show here before I go back into the presentation, tell you a little bit more about what's behind the scenes here, is as well as the general collection of data where we've taken most of what we've got and done the best job we can at the moment converting it, we have a much more tightly curated subset of the data from what's called volume electron microscopy, which is a family of techniques involving serially sectioning samples, imaging sections, and then gluing them back together computationally, which is quite an important emerging area. We wanted to do this because it was a good way to test large-scale 3D imaging and a good way to test curation processes, because one of the things that's critically important to get right in this data to even be able to visualize it properly is voxel spacing. So if you don't get voxel spacing right, all sorts of bad things will happen when you try to, certainly when you try to measure it, but even when you try to look at it. So we had to develop tools and pipelines for as well as conversion for manual curation of that voxel data. And then in this case, we actually use a different viewer. So we use Neuroglancer because it's great for this kind of data, not surprisingly, given the sort of origin in connectomics, and connectomics is basically volume EM. So most people have probably seen this, but it's an amazing piece of engineering and does the whole kind of zooming out from single cell level down to organelle level, and even for our big Platinaris data set, you can go from organism down to organelle, which is pretty cool. And the remaining thing, and I'll show it now just because I'll come back to a little bit later on, is that we also have again, we have the metadata here. But there's an interesting question about where this metadata comes from. So this is, I kind of think of as being the kind of shiny tip of the iceberg. This is the kind of the gallery. This is the showing the data. Underneath that, and the thing we've really been working on for the last couple of years, underneath the small point of the iceberg, is a lot of infrastructure around transforming, converting, moving data, representing that in databases, providing an API, which at the moment is only consumed by the website to build the front end, but will eventually be consumable by everybody else as well. And most critically, underlying this with data models that actually let us link these pieces up together. last sort of shout out, so Kaya really nicely introduced OME-EBI, and I'm sure you'll hear lots and lots about it over the last few days. This is an absolutely fundamental building block of everything we do on the visualization side, because basically our visualization strategy is convert it to OME-EBI, put it in S3, and let the wonderful array of viewers take care of the rest, because that gets it out of our head. And it's also worth, again, flagging that last year's challenge has been great for moving things forward, particularly on the ZAR v3 or OME-EBI version 0.5 side, because actually the slide or the part of the website I showed you with very many different volume EM images, some of those are actually ZAR v3 because they just would not work on our storage systems in ZAR v2 because individual images would be 12 million objects. Other critical component of this, and the other thing we've had to spend a lot of time on, is the metadata side. And metadata, you may have heard about or hear more about this set of principles, guidelines, EMBL-EBI. What we've had to do to make that work in a practical database is take this really high level guidance about what should go into a metadata model, what you should capture about image, and make it work, pull out the components, put the links between the things so that we can represent that internally in databases, and so that we can actually answer the questions that people coming to the database want to ask, which is often, give me images produced by this kind of method, but it's also give me images which are annotations of other images, or as we might call them, segmentations. More on that right at the end. Something worth mentioning is that we kind of hang the OME-EBI, which we almost view as the binary component of this, off a general container object called the image. And the nice thing from our side about doing that is it lets us have, as well as all of the sort of experimental metadata, we can have actually, in some cases, multiple different OME-EBI versions of the same data, because viewer compatibility does not at the moment cross everything, but also because this lets us gradually and relatively gracefully move towards OME-EBI, to move towards ZAR v3, so OME-EBI version 0.5 or later, in a sense on our timescales, and migrate across storage systems relatively smoothly as well. So last part of the talk, I'll actually go back to what I promised in the title, which is to talk about scaling, because this is something that I have to think about a lot these days. I mentioned at the moment that we get about 1 submission per day. If you project forward growth curves, this will probably be 10 in 2 or 3 years. At the moment, we have about 50 million files, which doesn't have a direct correspondence with a number of images exactly, but it's the scaling's still there. In about 4 years, we'll probably have 1 billion. So a number of things, the number of challenges we're going to have to solve over those few years. And I've picked out 3, which I think from my side I see is the biggest. Notably, I'm not putting actual volume of data storage in this list because thankfully for various aspects of the way we're funded, the way we share infrastructure with the big sequencing resources, this is not a problem, at least not yet. The bigger challenges for us are thinking really about how we scale curation, how we scale complexity, and how we scale the deposition processes. And the deposition process is something that you have to care a lot about as a deposition database because you're getting data in from the outside world. You have to be able to do it quickly because often we're the thing between a scientist and their publication. That's not a good place to be for a long period of time. So we have to make it fast enough to meet community needs, but also get enough metadata to make things useful. And increasingly, people want us to support programmatic or at least semi-programmatic deposition so that people can kind of give us packaged data, often coming out of their image data management system, often, for example, a Mero. And also something that is now my problem is that we're running 2 big imaging databases, which are separate for historical reasons and also for different community reasons, but these need to have shared infrastructure, they need to have shared underlying models, things like that. But this also provides an opportunity because it's a chance to do some new things, and actually that's really what we've been doing. Where I think we're going, and this maybe came up earlier, and the other aspect of last year's OME challenge that was really interesting for us is it kind of got us hands-on experience of working with RO Crate, because that's a very promising way to package data and metadata, at least when it comes into us, not necessarily purely with OME-EBI, because we do need to take data that isn't NGFF at the moment. Second side of scaling is the curation side. And there's lots of places where curation can happen. It can happen, or mixing up in a sense curation annotation here, but basically in a sense adding things to the image metadata to enhance it. It can happen before it gets to us in the kind of pre-publication phase. It can happen as it comes in, so that's submission, and it can happen afterwards. So it can happen when we do annotation of the data once we've got it. And one of the things we've been looking at the last part of these, but I think is going to be critically important for all of this, is what AI, which is a very, very broad set of topics, I'm aware, can help with this. And in fact, I sort of flagged this earlier, but a lot of the metadata in the collection of volume EM data, because we actually never captured that initially because of the way that Empire's deposition system works. We went back to the papers, we pulled the information out of the manuscripts, formatted it into the different components that or at least our implementation of the EMBL model has, but we didn't do that by hand, we did that by LLM, which does at least the initial pass of this, and then we do some human verification of the results. And the good thing about that is that can scale. It's only solving part of the problems, and there are challenges, so this is almost one of the easiest examples we could have picked out, but at least under certain circumstances that's working for us. Final part, last couple of slides of the talk. And one of the biggest challenges scaling, I think, is complexity, because increasingly people do not just want to do just one modality of imaging, you want to do multiple modalities because, for example, you need to do correlative imaging to target something using light microscopy and then volume EM to actually go into much finer resolution of the thing you've targeted. But also you want to combine imaging with gene expression data, do transcriptomics, add in proteomics, add in metabolomics, and also do lots of complex workflows. And we're going to have to deal with that going forward. One of the ways in which we're doing that is we're looking at a pilot for integrated spatial transcriptomics data. It currently sits in 2 different places at EBI. And I was going to say we'll need to look for funding for this, but we have some very good news this morning that we now have a grant to start on prototypes for this work. And another example of something that's very much in mind at the moment as an example of the kind of really complex workflow that we have to be able to accommodate for depositing data and then getting information back is something called cryo-ET, so electron tomography, which is a big part of in situ structural biology, which is sort of a really critical imaging technique. And this is good because it's a real example of complexity that we're grappling with at the moment. So it involves taking samples, imaging them to produce what the usual term for these things is movies, because that's what the giant electron microscope produces. Those are then kind of split up into tilt series. Those are reconstructed into 3D tomograms, and then people annotate the tomograms with both combinations of point annotations and also volumetric segmentation. Some of these things are images, some of them are 2D, some of them are 3D. Point annotations are not images, segmentations are in a sense a different type of image. We need to be able to represent all of this, and we need to be able to represent the links, and people actually need to be able to query interesting pieces of information here. So what was the sample in this tomogram is something you absolutely want to ask, but there's different sort of stages in this process. This is why the work that we've been looking at internally on data models has been so critical. And basically what we've arrived at is a data model where images get produced by inputs and some kind of process with a protocol, which looks very abstract, but lets us solve the main sort of 3 challenges I think we have there. One of which is that images which are produced by microscopes are take biological samples with an acquisition protocol, which is where information about the instrument and acquisition would sit, and they produce images. There are segmentations, where actually in this case the input is another image. The protocol is either a human doing some segmentation or a machine, an algorithm, or some combination of the 2. And then processed images where the input is again an image, but actually what the process is some kind of usually algorithmic protocol for producing the output. And we glue those things together and we can basically get to what we want collectively for the model. So I am wrapping up there. There are many, many more things I would like to be able to say, but want to be able to stick to the time. So I'll just finish by thanking both the teams I manage now for the Bioimage Archive and Empire, and the funders who make all of this possible, and all of you for very kindly putting up with an online presentation and staying attentive. So thanks everybody, and I'm happy to try to take some questions if you have them.
One, maybe two questions. Katerina.
So hi, this is Katerina. Great talk. Regarding your more generalized model, it's very similar to me to the ISA model that like there is an assay that produces, so the process could be similar to the assay. Have you looked into that?
Yes, it would map a lot closer to the ISA model than things we've had before. And actually, this is a kind of a design feature, because periodically people say, hey, we're using ISA to manage all the rest of this data. Can we use it to submit image data? There are some differences because there are more image specialized features, but the parts that overlap, especially things like study, we would absolutely keep the same.
Okay, that's great. Thank you.
Yes, thanks, Matthew, for the talk. It's more a comment than a question for Matthew. I think you had a great slide where you had PFF as a pipeline replaced by NGFF. And I think to echo what Jason was saying, we still have to consider that bioformat is still a key component of that process in terms of the conversion for ZAR. So I think as a group, we need to consider. I know Glencore has taken now the daily testing, as Jason indicated yesterday, but it's going to be the CI part of that, the conversion pipeline, etc., is going to be still very critical for making that all that workflow possible. So I think that is probably going to be a sharing testing effort that we need to share among all members of the community in order to achieve what Matthew has shown, which is wonderful. But that is a key point, not to forget, I would say. And Jason said
absolutely, that's a really fundamental bit of infrastructure that we have to think about.
Yeah, it's a very difficult piece to fund. So I think say, okay, we're going to build CI for that, people will not fund that. So I think how we can share the load, and the previous presentation showed 1 million of files we have to test, and if we want to run that daily, this is going to be very painful. Just don't want to tap it. But it's a very serious point we need to consider from moving forward.
All right, let's thank Matthew again.